The overall goal of this set of experiments is to precipitate plant host cell proteins using heat, thus facilitating subsequent chromatographic purification of recombinant biopharmaceutical proteins.
This method can help to streamline process development and increase product stability, for example by inactivating host proteases. The main advantage of this technique is that it is easy to implement into existing processes and can be quickly adapted to different target proteins. Demonstrating the procedure will be Hanna Gruho, a master's student from my laboratory.
To heat precipitate host cell proteins, for example from tobacco leaves, after culturing tobacco plants according to the text protocol, set up an 8-liter working volume water bath in a thermally insulated polystyrene foam bucket. Transfer the entire assembly onto a magnetic stir plate and place a magnetic bar in the water bath. Then surround the stir bar with a non-magnetic support tile, topping the stir bar by approximately 1 centimeter, upon which a polypropylene basket will be placed later during the procedure. Add 8 liters of buffer to the water bath. Then place a 23 by 23 by 23 centimeter polypropylene basket on the support tiles, ensuring that the support does not interfere with the stir bar rotation and that the basket is fully submerged in the liquid. Then remove the basket. Use an adjustable thermostat to bring the water bath to 70 degrees Celsius. Wait at least 15 minutes after the desired temperature is reached to ensure the entire assembly has reached a thermal equilibrium.